All right, here we are. Another day of Physiology Madness with Dr. H. Today, we're going to talk about some more endocrinology. We're going to be talking about insulin, glucagon, talk about their secretion, the stimuli that cause them to be released, and their overall function. And yes, we're going to do them together, both insulin and glucagon together. In order to get started here, we need to know what organ insulin and glucagon are secreted from. And the answer to that, hopefully most everybody already knows, is the pancreas. So we're going to start here with the pancreas. And the pancreas is what's known as a heterocrine. A heterocrine. It's a heterocrine gland because it secretes more than one type of hormone, and it secretes exocrine hormones and endocrine hormones. So pancreas has an exocrine function. This is the large majority of the function of the pancreas. This is like over 98%, 99% of all pancreatic function is as an exocrine. And it's good to know that. It's good to get this kind of out of the way. The exocrine function is due to the presence of acinar cells. There's also another cell called a duct cell. But acinar cells, and the acinar cells, or acini, these cells release digestive juices, stomach juices. All right, so they're not stomach, digestive juices, intestinal juices, pancreatic juices. These are the ones that are going to go into the duodenum, into the small intestines, and they're going to help with digesting the food coming from the stomach. So these acinar cells, they secrete digestive juices. Bunch of enzymes. That's a bicarb-rich solution. Okay, great. That's out of the way. You guys will learn more about this later when you start getting into the digestive system. The other cells of the pancreas are called endocrine, right? So the pancreas has both an exocrine function and an endocrine function. And the endocrine function, this is what we're after. The cells that are of endocrine function inside the pancreas are known as the islets of Langerhans. The islets of Langerhans are also just known as pancreatic islets. And that's kind of what we're going to go ahead and refer them to. These are going to be pancreatic islet cells. Pancreatic islet. Islet as in island. Because if you look at the histology of these, they look like little islands within a sea of purple. So the pancreatic islets. And the pancreatic islet cells. Now, we're starting to get into the nitty-gritty of this. There are two major pancreatic islet cells that we're going to focus on for our today's study. Okay? One of those pancreatic islet cells is called a beta cell. And the other one is called an alpha cell. The beta cells, these are the cells that secrete insulin. So as we go to continue our flow chart, we can go ahead and draw a little arrow over here, and we're going to say the beta cells, if they are positively activated, are going to secrete insulin. Whereas the alpha cells over here, these alpha cells, these, if they are positively activated, are going to secrete glucagon. All right, so now we know what secretes insulin and glucagon. What we need to look at next is what are the stimuli. All right, so beta cells are activated by a few things, but the primary thing that activates beta cells, the number one thing that activates beta cells, is an increase in blood glucose levels. BGL. An increase in blood glucose levels. And this increase in blood glucose levels, this is going to cause a humoral stimulation of the beta cells, and we're going to look exactly how. But this increase in the blood glucose levels is going to end up activating beta cells and causing the secretion of insulin. Alternatively, for alpha cells, well, the stimulus for the release of glucagon is now going to be a decrease in blood glucose levels. So anytime there is a decrease in blood glucose levels, this is going to go and talk to the alpha cells, and it's going to stimulate those alpha cells to secrete glucagon. There are other stimuli for both glucagon and insulin. We'll talk about them in just a minute. But I want to get it out of the way up front and early here is that insulin and glucagon are antagonists. So what this means is if blood glucose levels are high, we want to secrete insulin. Well, at the same time, we actually want to inhibit glucagon release. And it's directly inhibited by this increase in blood glucose levels. So as blood glucose levels are increasing and increasing, they're going to go over here and talk to these alpha cells, and they're going to turn them off. At the same time, when there's a decrease in blood glucose levels, I shouldn't say at the same time, alternatively, if there's a decrease in blood glucose levels, that's now going to go over to the beta cells, and it's going to turn them off. So you can see they're antagonists. When one is high, the other one is low. They're always working as opposites. They're brothers. They fight to get a job done. All right, so now let's go over to insulin here. And let's look at how insulin is actually secreted. So in order to look at the secretion of insulin, we're going to look at this box right here. This box is going to allow us to understand the secretion of insulin. So up here, secretion. Of insulin. Well, in order to understand the secretion of insulin, I think the first thing we need to be able to do is understand what classification of hormone is insulin. Insulin is large and in charge. Therefore, insulin, created by these beta cells over here, is a protein. Now, what do we know about big polar proteins? Is that they are water soluble, not lipid soluble. And so as this cell makes insulin, can it just leave this cell on its own? The answer is no. This cell, this beta cell right here, as this beta cell makes insulin, it has to package that insulin inside of vesicles. That's that whole nucleus, transcription, translation, goes to the rough endoplasmic reticulum, and the ribosomes make the protein, and the protein is going to go to the Golgi apparatus. And in that Golgi apparatus, all of these proteins are going to be packaged inside of vesicles. So now we have insulin that's been synthesized by this cell, packaged inside of vesicles. In order for these hormones to be released, these vesicles are going to have to go through something called exocytosis. They're going to have to get transported to the cell membrane. And just as a reminder or review, or maybe learning for some of you, is that there is an ion, a secondary messenger, if you will, that is essential for exocytosis throughout the entire human body, and that is calcium. So there's a story that we can talk about here, is that we know proteins are stored inside of vesicles, and they need to go through exocytosis to be released. And that exocytosis is going to require calcium. So we already know the end of the story. What do we know? Is that if beta cells are stimulated by an increase in blood glucose levels, that's going to cause the release of insulin. What secondary messenger must increase inside of beta cells? calcium. And that's going to drive exocytosis of insulin. So how does that happen? Let's look at that. Well, when there's an increase in blood glucose levels, so here inside of the blood, there is going to be more blood glucose, and the glucose is being transported through the entire body. Now, there are transporters found on the cells of your body, especially these beta cells, that are going to allow for glucose to go from an area of high concentration to low concentration. It's going to allow for diffusion. It's going to help for diffusion. And we have a facilitated diffusion transporter here called GLUT, G-L-U-T. And on these beta cells, it's a specific isoform of GLUT. It's called GLUT2. And GLUT2, these things are always found on the cell membrane. No matter what, they're going to be found on this cell membrane. And again, they allow for glucose to go from areas of high concentration to low concentration. So let's think about that. As our blood glucose levels increase, glucose is going to be able to go from an area of high to low. From high to low. And it enters this cell from an area of high to low. Hence why high blood glucose levels causes glucose to start pouring into the beta cells. What do the cells of our body do with glucose? Well, they convert it to ATP, right? They're going to go through a process of glycolysis. They're going to turn it to glucose 6-phosphate first, and pyruvate, and put it through the acetyl-CoA and then into the Krebs cycle, and it's going to make ATP. Now, if you followed all that, great. That means that you know biology. If you didn't, maybe go study that part. But for this, what I really want to get at is as glucose levels increase inside of this cell, there's more glucose inside of the cell. Well, this glucose is going to go and get made into ATP. So now there's more ATP. Sweet. ATP is used for energy. Lots of cool things that ATP can get used for. But we have another really interesting little transporter over here. Okay, this is a gated channel. And more specifically, it's a gated channel for potassium. So it's a potassium channel. But this particular channel is sensitive to ATP. When ATP levels are low inside of the cell, this channel is going to allow for potassium to kind of freely move out of it. So let's see, where is potassium highest? Inside the cell or outside the cell? Potassium is always higher inside the cell. So let's go ahead and draw some potassium in here. All right, we got some potassium. My son's been using my markers for drawing. Starting to die here. So potassium's higher inside the cell. Doesn't mean there's no potassium outside. There's just less of it. All right, so now if this channel is normally open, potassium is going to be leaving the cell because potassium is going to move through this channel from an area of high to low. So potassium would normally be leaving. And as positive charges leave the inside of the cell, does it become more positive or does it become more negative? Well, the positive charges are leaving, the inside of the cell becomes more negative. And we call this hyperpolarization. And as long as it's hyperpolarized, there's not electrical activity going on inside of the cell. However, this particular channel is sensitive to ATP. So let's write that down. This particular channel is called an ATP sensitive potassium channel. And as long as ATP is in the system, it is going to go down and it's going to bind to this channel and it starts to close this channel off. And potassium does not get to leave the cell as fast. However, something that is always in the surface of every cell of the human body is a sodium potassium ATP pump. So let's draw in a pump over here. This is a sodium potassium pump. We don't want to ever forget about them. They often get left off of pictures, but we don't want to forget about them because they're always active. And they're using ATP, so they are still pumping sodium out of the cell. 3 sodium out, technically. and they're pumping potassium into the cell, 2 potassium in. So here we are in a scenario where potassium is no longer going from an area of high to low. Potassium is not leaking out of the cell anymore. Instead, it's getting left inside of the cell. Not only that, but this pump is pumping potassium at the same time. And what's that going to do to the inside of the cell? It's going to start to cause depolarization. As the concentration of potassium inside of the cell increases, as the concentration of potassium inside of the cell increases, this is going to cause depolarization. And that depolarization process means that there's more plus charges building up inside of this cell. The last part of this story is, what are these plus charges going to do? What are all these positive charges going to do? Well, I got one more channel over here. This channel over here is a voltage-gated calcium channel. A voltage-gated calcium channel. This is a channel for calcium. As long as it's open, calcium can go from an area of high to low. Where is calcium higher? Calcium is highest outside the cell. Lots of calcium outside the cells. Lots of calcium outside the cells. Lots of calcium outside the cells. Very little calcium inside the cell. So as long as this channel is open, calcium is going to be able to go from an area of high to low, and it would enter the cell. But this thing is normally closed. Therefore, calcium levels inside of the cell are normally low, and therefore no exocytosis. However, when there's depolarization, these plus charges, that's voltage. These plus charges are going to come over here, and they're going to be activating. All these plus charges are building up on this plasma membrane, and they activate this voltage-gated calcium channel. Therefore, calcium enters the cell. And what's going to happen to the concentration of calcium inside the cell? It's going to start to increase. And this calcium, the secondary messenger, now is going to talk to these vesicles. It's going to bind the vesicles to the cell membrane, and it's going to drive a process called exocytosis. And now we have insulin that's getting exocytosed. I love that word, I make it up. But insulin's going through a process of exocytosis, and that is going to start to release insulin into the blood. Now there's an increase in insulin release. It's quite the process. We start from scratch, and we draw it out to understand it better. One more time. When there's an increase in blood glucose levels, that's the stimulus. Glucose starts to enter the beta cells through GLUT2. As glucose enters, it's going to go through a process of getting converted to glucose 6-phosphate. Glycolysis happens. We got pyruvate. We got acetyl-CoA. And it goes into the Krebs cycle, and it's going to make more ATP. ATP, the more of it inside of the cell, the more ATP is going to bind to the ATP-sensitive potassium channels, thus closing them. If the ATP-sensitive potassium channels are closed, potassium cannot leak out, and it's going to start to build up inside of the cell. As it builds up inside of the cell, positive charges build up inside of the cell, and we call this depolarization. The depolarization is going to drive the opening of voltage-gated calcium channels, allowing calcium to diffuse into the cell, increasing calcium concentrations inside of the cell, and allowing for exocytosis of insulin. So now we have insulin in the blood. A lot of work to get insulin into that blood. Now, let's communicate about what insulin does. What are the target organs for insulin? Flowchart. One target organ for insulin. The first place we want to take it to, well, if we're dealing with glucose, we got this wonderful organ right here called the liver. Liver, always the bridesmaid, never the bride, does so much work, gets very little credit. We're going to be saying that that has receptors for insulin, and it does. What else has receptors for insulin? All right, Next location we're going to go to is, let's take a stop at fat cells. Adipose. The last stop that we're going to take is skeletal muscle. There are other targets for insulin throughout the body, but we're going to go ahead and concentrate our attention on these three things. In the liver, what's going to start to happen in the liver? So in the liver, this is where glycogen is synthesized. This is where over 98% of your blood proteins are synthesized. And when is your blood glucose levels high? After you've eaten a meal. And if you have a ton of extra glucose, what do we want to do to all that glucose? We want to use it, and we're going to use it. As blood glucose levels are high, let's use that glucose to make ATP right now. And then let's take that ATP and let's start making things. And the liver is no different. So what the liver is going to do is the liver is going to have an increase in glucose uptake. So the first thing that's going to happen is that the liver also has GLUT2. If I have GLUT2, then glucose can go from an area of high to low. So in the liver, we're going to have an increase in glucose uptake. And that glucose uptake is happening through GLUT2. Now, as glucose levels increase inside the liver, the glucose goes through the same process as happen in these beta cells of the pancreas. And that glucose is going to get converted into ATP. So now we have more ATP inside of these cells as well. So as we think about the glucose uptake, let's also think about the fact that this is going to increase ATP inside of the liver. Now we have more energy to do things inside of the liver. What can we do? All right, with this extra glucose, let's store some of it away. Let's take glucose and polymerize it into big long chains of glucose. And what is that called? Glycogen. And in making new glycogen is called glycogenesis. So as we take this energy and now we start storing excess glucose away as glycogen, well, the liver does that in excess and we have glycogenesis. What else? Well, in order to understand the next thing, let's look at another stimulus for insulin secretion other than blood glucose levels. And that is amino acids. As amino acids increase, say for instance you just ate a protein diet, you just ate food with lots of amino acids. These amino acids in the blood, they also are going to stimulate beta cells. They do it through another mechanism that you don't have to learn about, but they're going to stimulate these beta cells to secrete insulin. So now we have more amino acids in the blood. That's a stimulus. And what do we know about the response? The response is always the opposite of the stimulus. So what else do we have going on over here? Not only is the liver acting on glucose, but it's also going to act on amino acids. Insulin is going to activate a transporter for amino acids, and it's going to increase amino acid uptake. Uptake. Remember, it's that fancy word for removing amino acids from the blood, putting them into the cells. So now I have more amino acids inside of my liver as well. Well, if I have more ATP and I have more amino acids, what does the liver make a boatload of? All of those proteins. Huge abundance of different types of proteins. So what can it do with this excess amino acids and this excess ATP? Let's make some proteins right now. So now we have more protein synthesis. And insulin is going to drive DNA transcription, translation, and protein synthesis. So these are all happening in the liver. Next, adipose and skeletal muscle. So now in order to understand adipose and skeletal muscle, let's come over here. So far, we've talked about glucose uptake through GLUT2. But there are other GLUT transporters. These allow for facilitated diffusion. There's GLUT1, GLUT2, GLUT3, GLUT4. It goes all the way up to GLUT14 as far as I know. There could be even more now. GLUT2 is known as insulin independent. GLUT2 is always in the cell membrane. GLUT2 is always in the cell membrane. But there are GLUT transporters that are not always in the cell membrane. There are GLUT transporters that are sometimes stored inside of the cell. GLUT transporters, well, all membrane-bound transporters are proteins. Now, what do we know about proteins is that they are polar and they are water-soluble, and therefore, in order to exit the cell or integrate into the cell membrane, they're going to have to go through a process of exocytosis. I hope this is starting to make sense. We're starting to see this so many times. So now, what if I had a transporter that was integrated in a vesicle? This transporter is going to have to be transported through a process of exocytosis to the cell membrane to be integrated into that cell membrane. Okay? These transporters here are known as GLUT4. And GLUT4 are insulin dependent. GLUT4 are a particular type of GLUT transporter that are insulin dependent. That means they require insulin for them to be active. Right now, if they're inside of the cell, they're not active. So we need to somehow get them from here to there. Well, again, what process is going to have to happen? Exocytosis. And what secondary messenger is going to have to be activated? Calcium. So this process of exocytosis is always driven by calcium here. Well, how does this all work? This works because on the cell membranes of adipose tissue and skeletal muscle, we have insulin receptors. Okay, these are IR, standing for insulin receptor, and they're a type of tyrosine kinase receptor. And for my class, y'all don't need to know everything about tyrosine kinases. If you're in somebody else's class, you might have to know about PI3K and AKT and how that's going to end up stimulating calcium. But for me, an insulin receptor, this is a tyrosine kinase. So inside the cell, there's going to be a chemical reaction. So now I have an insulin receptor here. And in the bloodstream, we have insulin that has been released. So in the bloodstream, insulin is flowing through the blood, through in the plasma, and this insulin is going to find these insulin receptors, bind to them, and activate them. And when this thing gets activated, with an arrow up, activation, when this insulin receptor gets activated, this is going to end up triggering a cascade response. Again, for those that need to know, PI3 kinase, AKT, but ultimately, it's going to end up causing an increase in calcium release inside the cell. Not through the same process as up here. And increase in calcium. And this calcium now goes down to these vesicles and ends up causing exocytosis. Once we get exocytosis of GLUT4, now these membranes are going to be covered in GLUT4. GLUT4 is a glucose transporter that is going to allow for glucose to go from an area of high to low. Where do we have an increase in blood glucose levels? Out here, right? There was more glucose here in the blood. That was the trigger for insulin secretion. So now that GLUT4 is here, this glucose that was in the blood starts to pour into these cells. Now that GLUT4 has gone through exocytosis, that causes an upregulation of GLUT4, glucose is now going to be able to rush from the blood into the cells. This happens in both adipose tissue and skeletal muscle. So over here in adipose tissue, we're going to have an insulin function on adipose tissue, which is to increase GLUT4. I need my markers. Goodness gracious. Increase GLUT4 upregulation. And by having an increase in GLUT4 upregulation, what this is going to do is it's going to allow for an increase in glucose uptake. More glucose is pouring into that adipose tissue. What does adipose tissue do? Adipose tissue is long-term energy storage. What's stored in adipose tissue is triglycerides. Tri meaning three fatty acids. Triglyceride, the glyceride being a glycerol. So it's 3 fatty acids bound to a glycerol. And if we have excess glucose in our blood, we are going to try to use as much as we can and store the rest away. What can our body actually convert glucose into? Our body can convert glucose into 2 things. 1, glycerol. We're going to increase glycerol synthesis. 2, we can actually convert glucose, portions of glucose, into fatty acids. Increase fatty acid synthesis. And then the adipose tissue can take that glucose and it can take those fatty acids, could take these 2 things right here and combine them. And when it combines these, now we're going to be increasing fat production, making new triglycerides. What is this called? Lipogenesis. As we make new fat out of glycerol and fatty acids, this process is called lipogenesis. So in adipose tissue, insulin is going to directly allow for glucose uptake, and it's going to trigger some enzymes that start to make new adipose tissue. Skeletal muscle. Not too much different than adipose tissue. Skeletal muscle also has these insulin receptors, and that is going to allow for an increase in GLUT4, insulin-dependent glutes. So now we're going to have an increase in GLUT4 upregulation. This increase in GLUT4 upregulation is going to allow for an increase in glucose uptake. Just like everything else, more glucose inside the cell, we're going to have more ATP. Awesome. Insulin, also in skeletal muscle, similar to the liver over here, is going to cause an increase in amino acid uptake. What can we do with amino acids? Turn them into proteins. So now we're going to take these amino acids and turn them into proteins. So this is protein synthesis. Lastly, skeletal muscle has small stores of glycogen. The liver has huge stores of glycogen, but skeletal muscle has small stores of glycogen. It helps run skeletal muscle contraction. So insulin inside of our skeletal muscles is also going to increase glycogenesis. To a small degree, relative to the liver, but still valid. This is the general function of insulin. We can see there are 2 stimuli. There are actually hormones that can stimulate insulin. There are hormones that can turn insulin off. But the general increase in insulin secretion is due to an increase in blood glucose levels. We inhibit it by decreasing blood glucose levels. And when blood glucose levels increase, what do we know the body wants to do? bring them back down. Take the glucose and pack it into the liver, make it into glycogen. Take the glucose, pack it into fat cells, make new triglycerides. Take the glucose, put it into skeletal muscle, and start making more muscle. The other thing that typically increases are also amino acids. Hopefully we're doing it together with a healthy, balanced diet. And those amino acids go to the liver and produce new proteins. Those amino acids go to skeletal muscle and make new proteins. That's insulin. Glucagon is actually not too difficult to learn because it is the opposite of insulin, but much more simplified. Glucagon has two primary organs that I ask my students to learn about. Okay, the first organ is adipose tissue that we'll write in here. And the second organ that we're going to discuss is glucagon's primary target, which is the liver. Always the bridesmaid. Let's go ahead and start here in adipose tissue, though, because I think it's important to think about. Okay, the primary stimulus for glucagon secretion is a decrease in blood glucose levels. So what are we going to want to do? We're going to want to increase blood glucose. Well, adipose tissue doesn't have glucose, but it does have triglycerides. And those triglycerides can be broken back down into glycerol and fatty acids. So adipose tissue responding to glucagon, well, it's going to activate some hormone-sensitive lipases that are going to start to cut up triglycerides. If we break down triglycerides in adipose tissue, we call this lipolysis, right? Lipolysis. So as we increase lipolysis, that's going to start to release glycerol. And it's also going to be releasing fatty acids. Fatty acids, some of these fatty acids are going to go into Krebs cycle. Some other ones, like odd chain fatty acids, can actually do the same thing that glycerol is about to do. Glycerol, check it out, check it out, check it out, is going to go to the liver. These odd chain fatty acids can go to the same location. But in the liver, this glycerol can get converted into glucose. What's it called when we take something that was not glucose and make it into glucose? We call it gluconeogenesis. I'm running out of room. Genesis. Gluconeogenesis. What's it called when we take something that wasn't glucose and make it into glucose? Gluconeogenesis. And that's going to now start to increase our blood glucose levels again. In addition to gluconeogenesis, one of the primary triggers of glycogenolysis is glucagon. So as glucagon binds to liver receptors, it's going to immediately start taking the glycogen, cutting it apart, and we call this glycogenolysis. Glycogenolysis, right? But glycogenolysis, that's how it's pronounced. So that, in conjunction with gluconeogenesis, these two things together are going to start to increase blood glucose levels fast, rapid increases in blood glucose levels. So you can see glucagon is a much more simplified idea than insulin. It's doing the opposite. They're antagonists of one another. Before we finish out, I want to actually add just a couple more stimuli to this process, just so you can think about these other stimuli. Say, for instance, with alpha cells, it's not just a decrease in blood glucose levels, but what if it's prolonged decrease in blood glucose levels? So something like starvation, like fasting, long-term fasting. That's going to trigger the release of glucagon. What else? Exercise. Exercise, as we exercise, ATP is getting created. to help with the energetics of exercise. And in order for all that ATP to have been produced, we needed to use blood glucose. And so our blood glucose levels are dropping as we are exercising. And as those blood glucose levels drop, the alpha cells are going to release glucagon. One more thing that's, I think, important to note in today's day and age is that some diets can cause glucagon to be secreted. One of the major diets that is going to act on glucagon is a ketogenic diet. Ketogenic diet. Because these diets, they are focused on not eating carbohydrates. And so if we are still eating, but we're eating less carbs, that's going to trigger glucagon release. Over here for beta cells, there's really just a couple more that I want to add. One is that when you ingest food, it's going to cause your stomach and your small intestines to start secreting hormones. Some of those hormones will come back into play later. But there are hormones like GLP-1 and GIP, okay, glucagon-like peptide 1 and gastro-inhibiting peptide. These hormones are produced in your duodenum, and they will go and activate beta cells. I just want that to be in your brain somewhere. One big more part, and then we'll be done here, is that we don't always want to talk about what's activating things. We also want to talk sometimes about what's inhibiting things. A major inhibitor of beta cells inhibition, a big minus right here. Sympathetic nervous system. So if we have epinephrine and norepinephrine in the system, that is going to inhibit beta cells. And at the same time, it's going to activate these alpha cells. Because if we're running from Mr. Axeman, this is adrenaline over here. If we're running from Mr. Axeman, do we want more glucose to be released, or do we want to store glucose away? I hope the answer is simple. We want to not store it away. We want to block storing glucose away. And we want to stimulate the release of glucose. With that, we have covered insulin and glucagon function. This is normal, healthy function. What would happen if things broke? What would happen if beta cells broke? Somebody had an increase in blood glucose levels, beta cells no longer secreted insulin, all of this wouldn't work. Again, if there was an increase in blood glucose levels, the beta cells could not secrete insulin. Now all of a sudden, insulin can't bind to insulin receptors. The person secreting insulin has stopped. No more insulin. Beta cells are broken. So insulin can't bind to insulin receptors. They have insulin receptors, just no insulin. And therefore, none of this exocytosis occurs, and all of this GLUT4 upregulation, GLUT4 upregulation in adipose and skeletal muscle tissue, it just doesn't happen. There's massive glucose uptake in the liver. It doesn't happen. But they still have insulin receptors. So when their beta cells are broken, can you give them insulin? Absolutely. Would they still respond to insulin? Absolutely. This is called type 1 diabetes. And in type 1 diabetes, the beta cells are destroyed in an immune system response, autoimmune reaction. And those type 1 diabetics are known as insulin dependent because they still have the receptor, and if we give them insulin, they will still respond to it. How about in type 2 diabetes? These are known as insulin independent because what's broke in a type 2 diabetic? It's not the beta cell, they're still secreting plenty of insulin for the most part. Rather, it's the insulin receptor. If we start to desensitize and downregulate this receptor, it starts to go away. So what's going to happen to the sensitivity of the cells of your body to insulin? If there's less insulin receptor, the sensitivity goes down, and therefore adipose tissue and skeletal muscle stops its glucose uptake. And that causes the glucose in the blood to remain in the blood. And that causes an increase in blood glucose levels, which is diabetes. So that's type 2 diabetes. And I just wanted to quickly introduce that to y'all here at the end of this lecture. So I hope you found this lecture useful, good learning experience, and feel free to like and subscribe if you want. You don't have to. I don't really care. I just want people to learn. So until next time, humankind, be both, please.